Proteins said as like the workers of the cell. They do a bunch of different roles, which we're going to talk about in the coming slides. And because they do a bunch of different roles, they have a bunch of different structures. A big theme in biology is structure and function, which we'll talk about many different times throughout the year. So we're going to learn about some different structures proteins can take, and that's going to relate to their function. If you were to like take an organism and just like remove all the water from it, and then you have all the molecules other than water that make up something living, half of that mass would just be proteins. So that's what I mean by like they are the main worker in the body, in cells. They do tons of different things, such as defense, storage, transport, cellular communication, movement, and then structure and support. So we'll go through each of these kind of briefly, one at a time. First one is enzymatic proteins. So we'll talk more about enzymes later in the year, but remember enzymes, well, let me back up. Think of a cell as just basically like a bubble, a lipid bubble, like a cell membrane full of chemical reactions. That's really all a cell is. Organized chemical reactions. That's why like you need to understand chemistry to really understand biology at a deeper level. And so all the chemical reactions that need to happen in the cell, if you didn't have enzymes speeding up those chemical reactions, they wouldn't happen fast enough for life to be sustained. So enzymes are like this little purple thing. Basically, they're kind of like a matchmaker. They bring the molecules that are going to be reacting together, and they make it happen quicker. Reactions could happen on their own, but the enzyme makes it happen more efficiently, like a job recruiter or something. Like in digestion, you have enzymes that are going to hydrolyze, going to break down your food. Right? When you eat your food, they're in the polymers, and you're going to break it down into the monomers. Storage proteins. Think of eggs, right? You ever like to just eat eggs? I like eggs, pretty good. But the idea of what's going on inside of an egg is you're going to have a protein code called ovalbumin. Definitely albumin. I'm not sure how we pronounce that, but anyways, it is a protein that's like a storage protein in the egg. If this egg were to develop into like the actual chicken, the embryo would start breaking down the storage protein into the individual monomers, the amino acid that's the monomer of proteins that the embryo would need to start growing. Okay, but if we just eat the eggs before they had a chance to develop, then well, you just get the benefit of the protein. Cool. Defense, especially like after COVID and everything. You heard a lot about antibodies. We got to get the antibodies against this virus. Antibodies are actually proteins, interestingly enough. And those proteins are specific to a certain virus or to a certain bacteria. So think of how many different viruses and bacteria there are out there. There has to be a specific antibody to attack any one virus or bacteria. Transport proteins. So when we talk about cell transport later in the year, we're going to learn about how some things can just go through the cell membrane on their own. Call that just simple diffusion. Other things need a protein to either facilitate the movement through or learn about active transport. You're going to need proteins that like essentially use energy to bring things inside the cell. So proteins are really important with transport. Okay, any questions so far? Again, as you get throughout the year, we'll get into more into these details, but just kind of going over some general functions. Hormonal proteins. Hormones, turns out hormones are actually, not all hormones are proteins, but a lot of them are proteins. Anybody know how you know like insulin is a protein? It's actually how you know a lot of proteins, things are proteins. It's kind of like a code word. It's at the end. IN, bless you. IN. So if you see words in an IN like casein, albumin, insulin, that's a sign, hey, that's a protein. Actin, myosin, so that's going to be a sign, hey, that thing's a protein. So anyways, the example of insulin, you might hear people with diabetes having issues with their insulin. Insulin takes the sugar from your blood and it will allow your cells to bring the sugar inside the cells so the cells can use that glucose. Okay. There are contractile and motor proteins, right? Like, you want to like pick up stuff or something, or like move around, or just be strong or whatever. You need proteins that will like pull on each other. Actinomycin, they almost act like Velcro. Have you ever seen like Velcro zoomed in? It's like little hooks and then loops, and they like hold on it. You know what I'm talking about? You ever zoomed in on Velcro? That's kind of how actinomycin are. It's really how your muscles are. Like you're like a bodybuilder with giant muscles. You just have like more massive pieces of Velcro. You got way more hooks and attachments going on. Cool. Receptor proteins. So in your nervous system, when one brain cell wants to communicate with another neuron, you are going to release certain signal molecules, some of these being proteins, and then those are going to bind onto a receptor protein that's on the cell membrane of a nerve cell that's next to another nerve cell. Cool. Structural proteins like collagen, keratin, right? You're talking about your hair. You want to keep your hair nice and strong. You want to support that keratin. Stuff like that. Or like, people start getting older, It's like, just look in the mirror, it's like, where did that wrinkle come from, You're just like, well, that collagen that makes up, or the connective tissue of our skin, that collagen starts breaking down, and then, you start just looking older, for example. Anyways, so getting into, I mentioned enzymes. I do want to show you a graph of what enzymes do. This should be familiar to you from chemistry. Remember these graphs, right? A little exothermic graph. And we said reactions go from reactants to products. But you first have to meet the activation energy. You have to invest energy first to make a reaction happen. And that's what I was saying earlier, where like without enzymes, you'd have to invest too much energy. It would take you too much time to keep meeting that energy investment, and you would just die. You wouldn't be able to have cells that could actually function. But enzymes lower the investment of energy you need, so you can do these reactions more often. Okay. They say without being consumed in the reaction. In other words, we can use enzymes multiple times because proteins are very expensive to make. When we talk about protein synthesis, you'll see it's a lot of steps that go into it. So that's why you want to keep being able to reuse proteins. Otherwise, you'd be having a separate issue. You'd be using all of your energy to just make enzymes, and it wouldn't really like actually help you out anymore. Okay, cool. All right, so the theme we've been talking about with the biomolecules is monomer makes the polymers, right? Monomers are the individual building blocks. Polymer is the bigger thing that you make. So the polymer for proteins are the polypeptides. So a key thing you need to be able to do in this unit is tell me what are the monomers, what are the polymers for these things. Polypeptides, poly means many, like we mentioned before. Peptides is referring to a peptide is an actual chain of amino acids, which you'll see is the monomer of the protein. Anyways, there are 20 different amino acids, 20 different monomers that proteins have. That makes proteins a little more unique, where you have a lot of variety. And then you combine those amino acids together in one way, they might make a structural protein. You combine together in another way, they might make like a hormone or something, a signaling protein. And then some proteins are made of just one polypeptide chain, which I'll show you more what I mean here in a second. Others require more than one. So here's the monomer of the of the polypeptide. Okay, the monomer is the amino acid, and we went over like the different functional groups at the beginning of this unit. This is where hopefully some of that investment of time starts to pay off a little bit. When you look at amino acid, you don't just like, it's not like you're just looking at a foreign language now. It's like, oh, that's an amino group. I'm familiar with what that is. A carboxyl group, I've seen that before, right? Think of the end of the fatty acid tail. They had a carboxyl group too. So you start seeing some of these similar building blocks. Now R is that can be changed out. So all of the 20 different amino acids have the same basic backbone, and then you swap out the side chains with something else, depending on what the amino acid needs to do, what the properties of that amino acid are. So how the amino acid behaves depends on the R group. And that's what you see here. These are the 20 amino acids. So fortunately for you, you do not need to memorize the amino acids. Now, if you take biochemistry in college, right, you keep doing biology, you will have to memorize all these amino acids. And if that like gives you a panic attack, it's right. I did it, okay? I don't know if that's like, well, that's true, but you can do it because it's all the same backbone. Only the highlighted thing, the color thing, is a different thing. Okay, and so you start to see certain patterns. Now, what I do want you to note are these categories, and specifically hydrophobic, hydrophilic. Super important with the proteins. Hydrophobic, water-fearing. So when you make a protein that has the hydrophobic amino acids, would want to be inside the protein to get away from the water. And the hydrophilic amino acids will be on the outside of the protein so they can interact with the water. Does that make some sense? So that's why you want to know hydrophobic, hydrophilic. Now, I have a question for you. What makes all these non-polar? Like, what is a common theme you see with all these non-polar amino acids? Because this seems overwhelming. It's actually a very, very simple thing you can look at to see, oh, yeah, those would be non-polar. Yeah, go ahead.
CH3.
Yeah, the CH3. So I told you, I don't know, back first class, if you see carbon to carbon or carbon to hydrogen bonds, those are non-polar bonds, right? The electrons are being shared more or less equally in those bonds. So that's why these amino acids are non-polar, because they are essentially ending up with a bunch of carbon to carbon and carbon to hydrogen bonds. Okay, there's some slight exceptions like methionine or tryptophan, but again, overall they behave in a non-polar way. Now there are like positive, negative charge, and then just more generally polar ones. I'm just lumping those in as hydrophilic. I think for your purposes, thinking hydrophilic. You don't need to memorize which ones have a positive charge or negative charge. Just know, hey, are they hydrophilic? So if I show you let me be clear. What I don't mean is, I'm not going to ask you tyrosine. Is tyrosine polar or non-polar? Tell me. I'm not asking that. If I show you the structure of it, if I show you that structure, can you look at it and be like, that's polar, that's non-polar? Basically, if I show you a structure, you see all these carbon to carbon, carbon to hydrogens in the ending in the R group, it's non-polar. But if you look here at the polar ones, see how like serine, yeah, it has the CH2 there, but it's got that alcohol group at the end. Same with threonine and tyrosine. Then you know that's polar. Or especially if you get the charge on it, plus or minus, then that's going to be hydrophilic because it can interact with the polar water. Questions on this? Okay. You want to start memorizing them? Go for it. All right, now that how do we make a polypeptide chain? So the monomer of proteins are the amino acids. How do we take the amino acids and make the polymer, the polypeptide? The same way we made all the other polymers. The dehydration synthesis. So you're going to, same kind of process here. You're going to take the hydrogen and then of one monomer, and then the alcohol of another monomer, remove that water out, and then boom, you got a nice covalent bond forming. Okay? Now, remember, you need to know the names of the covalent bonds that hold these polymers together. For proteins, it's the peptide bond. Peptide bond is just the name of the covalent bond that holds these monomers together. Okay? Technically, like, the amino end of one amino acid connects to the carboxyl end of the other amino acid. Cool, If you forget that, that's fine. Which, while I'm thinking of it, just naming terminology, the amino end has the amino group. They also call it the N-terminus because amino has the nitrogen atom. And the C-terminus is the carboxyl end because, I don't know, it has a C instead of an N. And then notice these different R groups in the yellow and the green. Okay? And then, again, all that same backbone going on. Questions so far? Cool. Okay, make sure I said everything here. So yeah, you're going to link them together with dehydration synthesis. Kind of like when we talked about the polysaccharides. If you just have two monosaccharides together, we call it a disaccharide. Same difference with proteins, except we don't say disaccharide, we say dipeptide. Peptide refers to proteins. Okay? And then polypeptide is just whenever you get three or more. And I think it's worth pointing out that these polypeptide chains can be like thousands of amino acids long. Like proteins can be very, very complex structures. And then again, you have the C terminus and the N terminus, carboxylin and amino N, and then those C terminus with N, C with N, C with N. Okay. Now, I was talking about the polypeptide. So once you've linked all the amino acids to the monomers and you made the polymer, the polypeptide, then what? Well, now you have what they call the primary structure of the protein. Okay, primary structure. They'll call it the balls on a chain, or beads on a chain. And then the secondary structure is when it starts to fold up. So you start to get this helix-like structure. And I have another slide that goes into a little more detail on this stuff, but just quick overview. You get a helix-like structure. Then if you get multiple secondary structures together, you make a tertiary structure. And then if you get some of the time, sometimes the proteins end right here. Other proteins, more complex proteins, you'll get multiple polypeptide chains together, and you then make the quaternary structure. So really the polypeptide chain is really whenever you get to that tertiary structure. It's really where people start like saying the word polypeptide chain. That's more the fully formed protein. Cool. This is just kind of showing you just how do you see proteins represented if you're like reading your book or you're just studying biology or something. There's a space filling model where they're just going to show them as like spheres. So this is just kind of helpful to get a general idea of like, hey, what is like the overall three-dimensional structure of the protein? If you want to get an idea of like, hey, what are the specific kinds of like secondary structures that make up this molecule? Because we'll learn about there's alpha helixes and then there's sheets. The ribbon model shows the sheets versus the helixes, helices, whatever. That's what the ribbon model shows. Wireframe model is helpful for like, if you're like, say you're researching, you're a pharmaceutical researcher, and you're trying to see, hey, how is this protein going to be able to interact with like a drug molecule? This is where that model is more helpful because you can start seeing these are all the R groups. You can kind of start seeing how they come out and interact with like, whatever your target is. I don't care if you had these memorized or anything. It's just kind of like an FYI. So. Some other ways you see it. If you just want to show like the most basic, like freshman bio, they're showing you proteins like this, where they just kind of just show like a blob, Maybe it's like literally just a blob, like a cylinder. They say enzyme. If you want to start getting a little more detail, so AP Bio starts seeing more of the structures like this, helping you see, hey, life is not all just like hexagons and whatever that is, right? And then, maybe like more like biochemistry, you'll start really getting into like, you can see some more of the secondary structures that make up that bigger protein. And then if you want to, if like they're trying to show a protein as like an export product especially, they'll just use like little dots to show like, oh, that's insulin or something like that. Cool. We'll keep going through 3.5b. All right, protein structure and function. The amino acid sequence, that's going to determine the overall three-dimensional structure, okay? So whatever amino acids make up the protein, that ultimately determines how that protein is going to behave. Because again, you had those different categories of amino acids like hydrophobic, hydrophilic. So depending on how many hydrophobic or hydrophilic amino acids make up the protein, that determines how it'll behave. And going back to like antibodies have a specific shape that can attack like a certain virus or bacteria. That's based on like the specific locations of all those amino acids. Okay, going in a little more detail, the 4 levels of protein structure. So I mentioned you have primary, secondary, tertiary, and then quaternary. This 4th level. Not all proteins get to that level. So let's run through those different structures. Primary, so it's just the actual just listing of all the amino acids. These 3-level abbreviations, those are the amino acids. So like GLY is glycine, this is proline, threonine. So this is just to kind of just quickly show it to you. And then you're assuming like, hey, you could look up the structure if you want to look up the structure. But I want to remind you that like, hey, all the amino acids have that same basic structure. What I do want you to know is that there is an amino end and a carboxyl end with that primary structure. Because you always have the amino end of one amino acid lining up with the carboxyl end of a second amino acid, you keep doing that on and on and on, you will eventually have 2 different ends of the chain. Because there's no option for like N to go with N or C go with C. Okay. Secondary structure. So there are 2 kinds of secondary structures. There is the alpha helix and the beta sheet. Okay. So the alpha helix is exactly what it sounds like. It's a helical structure. And why alpha helix? Well, alpha would refer to like one, like I believe alpha is clockwise, and then there's also a beta helix that would be, say, counterclockwise. The directions, of each one isn't so important, but what is important for both the alpha helix and the beta sheet are the hydrogen bonds here. So the benefit of these secondary structures is the hydrogen bonding. You begin to get a lot of strength that happens in the secondary structure because, like if I back up real quick, you get like, maybe amino acid 25, hydrogen bonding with amino acid, say 42. That's where like the hydrogen, like one part of the amino acid chain starts having interactions with another part. That's the idea of the secondary structure. So coils refers to the helix, folds refers to the sheet. Beta pleated sheet, it's literally exactly what it sounds like. It's just like a sheet. Think of a sheet of paper that's kind of like folded up. It will have all these kind of ridges going on. Okay, and then sometimes with the sheet they'll have like an actual head of the arrow, and that head points to the carboxyl end, and then the flat, or not flat, but the non-arrow part of the sheet end, that's the N terminus, the amino terminus. Good? Okay. All right, now tertiary structure. So once you take multiple of these secondary structures, like, all right, so you have an alpha helix, and then you combine that with the beta pleated sheet, and then those start interacting with each other. That's where the tertiary structure comes in. And what really starts happening in the tertiary structure are those side chains really start interacting with each other. So multiple R groups are starting to interact. Some of the interactions can be hydrophobic interactions. If you remember from chemistry, hydrophobic interactions, like van der Waals, or van der Waals, that's like it's basically like a momentary interaction that can occur between 2 non-polar parts of the protein. That's like like just for like half of a second, that part of the molecule might have a partial positive, so that creates a partial negative on the other part, on the part of the molecule right next to it, and that is a momentary attraction. But then it might fade away. But then some other part of the protein gets a partial positive, and that creates an attraction. If that's confusing, you don't need to know the details like that clearly, but just understand that there are hydrophobic interactions. There are these slight interactions that occur between the non-polar side chains of the amino acids. Okay? Not super strong, but just slight interactions. And then disulfide bridges. So if you look at the structure of the amino acids, some of the amino acids like cysteine will have sulfur atoms in there. And sulfur is pretty good at making covalent bonds. So if you end up getting like 2 cysteine amino acids that just happen to fold near each other, they can form these strong covalent bonds called disulfide bridges. So di means 2, sulfide referring to like sulfur atoms being bonded together. So, how are we doing on time here? I'm gonna leave it off there. We're starting to run out of time.